Okay, we're going to go ahead and get started. Welcome, everyone. Good afternoon, and thanks for joining us at the NINDS HEAL Biomarker webinar. My name is Rakanda Medley, and we will start off with a presentation of slides, followed by questions and answers. Please submit your questions only in the chat section featured below, and they will be answered at the end of the presentation. We ask that you please remain muted at all times. This presentation is going to be recorded and we will place it on our YouTube page. And I will be sending that link along with the presentation slides to everyone that has registered. I will now turn it over to our moderator for today, Dr. Ram Aruchichan.
Thank you, Rakonda. Good afternoon, and thank you all for joining the HEAL Biomarker webinar. I'm going to start sharing my slide and go over the video so that I could better see the slides. Slides coming up okay?
Yep, looks good.
Okay, thank you. So this webinar is mainly focused on this HEAL RFA, the discovery of biomarkers and biomarker signatures to facilitate clinical trials for pain therapeutics. And first, as an introduction to this HEAL biomarker team, so this is HEAL biomarker team. HEAL is the helping to end addiction for long term and also called as the HEAL Initiative. It's a trans-NIH effort to improve prevention and treatment strategies for opioid misuse and addiction and to enhance pain management. HEAL has over 31 distinct programs, and these programs are led by most NIH institute centers and offices. The HEAL Biomarker Program is led by the Division of Translational Research, National Institute of Neurological Disorders and Stroke, or NINDS. So this slide shows the team which is responsible for the biomarker program. I am the lead of the biomarker program, and Dr. Pelly Mounter, she is helping us as a consultant right now. She started the program and now she moved to the neurotherapeutics program. So she actually functions as the advisor for us. And then the program is ably supported by Dr. Silju, who is our health program specialist. And you now heard from Rakonda Medley, and she is the program coordinator and helping us to all the program activities. So before going into the RFA details, first few slides will be introduction slides about biomarkers. That would bring us all on the same page. So this slide shows the difference between the biomarker, clinical outcome assessment, surrogate marker, and the clinical endpoint. So we need to understand the difference between these two before we talk about the biomarkers. The clinical outcome is the measurement which measures patient's symptoms or conditions, and the surrogate marker is the one actually is being a substitute as a clinically meaningful endpoint, and the clinical endpoint measures how a patient feels or to function or survives. Rather, the biomarkers have distinct characteristics of objectively measuring and evaluating as an indicator of normal biological process, pathogenic process, or pharmacological response, and mainly to a therapeutic agent. So as this RFA specifically calls for response monitoring biomarker and response prediction biomarker, it is important for us to understand the difference between the category of biomarkers. So this slide shows the different categories of biomarkers and defined by the BEST. The BEST is a guideline put forward by FDA and NIH Collaborative Working Group. The BEST stands for Biomarkers, Endpoints, and Others Tool, and it's identified 7 distinct biomarker categories as indicated here. as diagnostic, monitoring, predictive, prognostic, pharmacodynamic response, safety or susceptibility risk. These are the categories of biomarkers. So to more understand this, that we need to know the specific context of use of each of these category of biomarkers. So what is the context of use? The context of use is the manner of use and the purpose of the biomarker. According to the best guideline, the diagnostic biomarker is a patient selection biomarker. And the monitoring biomarker, as it is indicated here, it detects the change in the disease status or indicate or find the safety and the exposure of a drug. And the monitoring biomarker, as it could be used as an indicator of toxicity or as a safety too. Predictive biomarker is identifying individuals based on effect from a specific intervention or exposure. Prognostic biomarker is mainly to stratify patients, and the pharmacodynamic biomarker could be used as the response biomarker, and it is an efficacy biomarker also, which could predict a surrogate endpoint, and also it is a response biomarker showing biological response related to an intervention or to an exposure. Safety is again a toxicity biomarker, and susceptibility risk biomarker indicates the potential for developing disease or sensitivity to an exposure. So it is important for the concept or the aspect of biomarker in context of use or the fit for purpose when it is being developed. So as here, this slide shows the biomarker categories according to the definitions and also how they are being used in the drug development. So this shows the different categories of biomarkers in the drug development process. And we only, for the interest of time, we are going to look into the biomarkers that are being more relevant to this particular RFA. So the monitoring biomarker is specially used either in phase 2 or 3 and clinical practice, which serially measure the status of a disease or the medical condition or for evidence of exposure to a medical product. And the pharmacodynamic or the response biomarker shows that a biological response has occurred in an individual who has been exposed to a medical product. And finally, the response prediction biomarker, which is another interest to us, shows that that serially assess the status of evidence of exposure to a medical product. So this slide shows the biomarker different categories in the clinical utility, and for the interest of time, just be look into the predictive biomarker, which is the one we are going to talking about more in the RFA. So the predictive biomarker is for a response to a treatment, and the time it's measured is before a treatment selection, and the clinical utility. So this is the context of use or the fit for purpose to identify treatments likely to be effective or guides the initial treatment decision making. So the points to consider when you write the context of use is identity, the aspect that is measured, and the form in which it is used for biological interpretation, and the species and the characteristic of the animal or the subject studied, purpose in the drug development, and the drug development circumstances for applying the biomarker, the interpretation and decision based on the biomarker, and the study design should address the context of use of the biomarker, mainly as what we have been talking about, the use statement, the very concise statement that should include the name and the identity of the biomarker and its purpose in the drug development, and the conditions for its qualified use. So how it's been used, that comprehensive description of conditions for the biomarker to be used in the qualified setting. Again, if you see the response monitoring biomarkers. So it should clearly indicate a therapeutic agent as engaged with the intended biological target and can be followed over the time, as well as the response prediction biomarker or signature. It should indicate that the individual is likely or not likely to respond to the therapeutic agent. So the response biomarkers are early indicators of efficacy in phase 2 or even phase 1b trials, and they are adjuvant endpoints in early clinical trials. And when used in combination with drugs exposure, they could be used to select dose levels in early clinical trials for acute or chronic drug studies. So the response prediction biomarker, the second category the RFA calls for, can stratify pain subjects into a homogeneous groups responding to a particular therapeutic mechanism of action. So this reduces the variability in clinical trial design. For example, if the therapeutic mechanism depends on increased expression of a particular voltage-gated channel, it will allow a more informed method for the development of this arm assignment. Likely most helpful in treatment decisions, allowing more accurate choice of therapeutic mechanism for the individual pain patient. So why we are only focused about the two biomarkers, prediction biomarker and the response biomarker. And this slide simply shows that in the drug development, the failure happens in the phase 2. And if you see that using the biomarkers and not using the biomarkers has been the success rate is almost half. So if you use the biomarker, the success rate is 46.3%. And if you don't use the biomarker, it is 23%. So this is a critical phase. And most of the response of predicting biomarkers could enhance these phase 2 clinical trials. That's why we are focusing mainly on these two biomarkers for this RFA, as well as this is the gap in the HEAL Biomarker Program, because for the response to the early RFAs, most of the funded projects are in diagnostic or prognostic biomarkers. So this just to give an illustration about how these biomarkers could be predicting, and this modality here shows imaging and where that in the brain the activation pattern compared to the drug arm and the placebo arm shows the differences. And you can use this in clinical trials to test a novel drug after validating that with the efficacy of a known drug. So this is what this RFA calls for, to finding a biosignatures or a biomarker for a particular therapeutic so that we can be using that in the phase 2 clinical trials for the similar trials. The biomarkers could be multi-component biomarkers, and this is a current FDA Prospective. So in principle, the similar considerations apply for any single component biomarker. I mean, like as you develop a signature or a single biomarker, the evidence for analytical and clinical validation should be there as proposed to the context of use. And how does the multi-component biomarker add value compared to a single component? And the rationale for inclusion of the different component and the weight of each of the different components when you combine them, and the importance of individual components as a whole to the whole signature, and the interpretation where one or more components may drive the overall result. So either the one component is important, or when you add up the other components, how they drive your overall results. And when you are using the machine learning, the method should consider the features of selection, and there should be a justification provided. As small differences in the input data can dramatically influence the output models, you should consider how critical are the specific assays that you have been considering for the machine learning, and also characterize the link to causal pathway and pathophysiology. So this has been taken out of a workshop that was organized by the FDA. The workshop happened last year, and for the identification of concept and terminology for multi-component biomarkers. And they are formulating a a white paper on this one, and that will be coming out soon, and we are actually waiting for their guidance on this. So the HEAL Biomarker Program, the main objective is to develop biomarkers and endpoints to facilitate discovery of new or non-addictive treatments for pain. And as the process involved 4 different phases: discovery, analytical validation, and clinical validation, and finally the FDA qualification. And the discovery phase include identification of the biomarker, establish that the biomarker is associated with the outcome of the interest, development of the detection method, conduct initial clinical validation studies. And in the analytical validation, you establish the performance characteristic for the biomarker detection method that is acceptable for the context of use, and establish the technical protocol or the process, and the clinical validation that being correlated to the concept of interest. And then the clinical validation study establish the association of the biomarker with the outcome of interest with clinical sensitivity, specificity, and the predictive values. And it should be classifying as predictors with the context of use, and evaluate overall utility for clinical practice and drug development. And then a large prospective clinical study should be included in the clinical validation also. And finally, this will lead to the FDA qualification. So FDA qualification is the review, FDA's review, and biomarkers allow specific and reliable interpretation within the specific context of use, and plan for integration in the clinical trial design and clinical practice. So for this RFA, these 3 phases have been covered. Not the final FDA qualification phase is not covered in this RFA. So you could propose a discovery of biomarkers, or if you have at that stage, then you could do an analytical validation studies or the clinical validation studies. And so the pathways we talk about that, saying that on the FDA qualification, and just briefly that not only the FDA qualification is for all biomarkers, and the biomarkers that you propose could be part of the IND or IDE or the NDA or BLA applications. And so those could be approved as an in vitro diagnostic device by FDA. And then we saw the biomarker qualification. And the third pathway that you could propose also the scientific community consensus, where you could get the CELIA approval. So this could be done in academic setting or in the lab setting also. So coming to this biomarker, particular RFA, so this RFA is discovery of biomarkers and biomarker signatures to facilitate clinical trials for pain therapeutics. So this is a UG3, UH3 mechanism, and we are going to find out that what it is. So this is a reissue of NS 22050. To develop, validate response monitoring or prediction pain biosignatures that will facilitate phase 2 clinical trials for pain therapeutics. And the web link is shown here. The application deadlines, the next application deadline is July 10th, and the other one is going to be October 23rd. As we talked about it earlier, the NIH initiative is a trans-NIH initiative, and the scientific context have been shown here, and this is in the RFA. And also what you need to understand is that HEAL has its own data sharing plan. So HEAL encourages the use of common data elements in all basic clinical and applied research, patient registries, other human participants of the research, to facilitate broader and more effective use of data advanced research across all studies. So this will be applicable along with other NIH guidelines for the data sharing plan. So UG3 UH3 phase award mechanism. So this is a cooperative agreement. In the UG3 phase, which is the phase 1, it is an exploratory or the development phase of the grant, and it could be up to 1 to 3 years. And the budget is limited to 500,000 direct cost per year. And the UH3 is phase 2, and it is an exploratory or the developmental phase, and it could be again 1 to 3 years, and the budget is limited to 1.5 million. So the total grant period is 5 years. So you could split UG3 and UH3, either one year UG3 and 4 year UH3, or two year UG3 and 3 year UH3, but together all this is a 5-year grant. And we intend to award 6 to 7 in 2023 and 3 to 4 awards in 2024. Awards issues under this NOFO are part of funds set aside to support the HEAL. So the main aspect of UG3, UH3 is this is a milestone-driven program. So going from UG3 to UH3 there's a go, no-go milestones. The milestones should be extremely clear, quantitative, and definitive milestones. And the transition occurs via administrative review. So the successful achievement of annual and UG3 transition milestones will be done through administrative review. The overall feasibility of project advancement considering data that may not have been captured in the milestones, and HEAL, and it depends on the HEAL programmatic priority as well as the availability of fund when the decision is made from UG3 to UH3. So the milestones are defined as scheduled outcomes in the project timeline that signifies the completion of major project stages or activities based on the parameters that are provided in the funding opportunity announcement. So the writing the milestones, that here are some examples. Then if you say the enrollment and recruitment will be completed by end of UG3 phase, that is not an acceptable milestone. So that should be clearly specified as how many subjects are going to be recruited and what percentage will be achieved in first year and how that will be done in the second year, etc. So here as an example, 1,200 subjects will be enrolled by the second year of UG3 phase, with 70% in the first year and 30% in the second year, and consistent with the power of, according to your study, what is the power is 85% of the p-value, as well as the milestone SOP and protocol development. But it should be clearly specified what is your SOP and what is the protocol development, like IRB approved clinical protocol ready by Q1, year 1. And then the assay reproducibility, as shown here, that should be defined as what kind of assay and then how many times it will be repeated and what sites will be conducting that, and the intra and the inter-coefficient variations, and equal to what percentage considerable as acceptable. So the milestones, if your project is going to be funded, the milestones will be established along with the program. As this is a cooperative agreement, according to each project, a scientific officer will be working with you to come up with the milestones. So milestones could address evidence that study planning and the team coordination is in place, that the evidence that metrics for clinical enrollments have been met, evidence that quality assurance parameters for sample collection or detection method performance have been met, the desired magnitude and reliability of the association between the biomarker and the target engagement has been achieved through an intervention in the preliminary studies, and the desired precision, accuracy, and the dynamic range of the biomarker detection method, and the initial prediction accuracy of the biomarker signature for response to the therapeutic. The transition from UG3 to UH3 phase. So complete the UG3 phase and submit all your annual research performance reports as directed in your notice of award. And the progress report can be submitted through ERA. Progress report must be approved by the program director. The transition milestone discussion between PI team and the NIH team will occur when you are ready to do that. And then the continuation of the UH3 phase will happen after that. So follow the instruction in your notice of award to apply that. So the application entry criteria. So mainly the application should focus on one pain condition or several pain conditions that have a common pathophysiology. Identification, assembly of a biomarker or biomarker signature, possibly a multimodal component that is being encouraged in the RFA, and strong clinical rationale. There should be a strong clinical rationale as well as a strong biological rationale. And the preliminary data, it should show the proposed therapeutic agents as robust efficacy, the development of a working hypothesis regarding the context of use, and how biomarker or biomarker signature will be used in a clinical trial, and plan for rigorous standardization of sample collection and detection methodology. And a multi-PI team is encouraged as the multimodality is being encouraged, and the knowledge and technical proficiency of the research team. So examples of response applications, identification and optimization of a single biomarker or a biomarker signature that can accurately monitor, indicate, or predict response to a specific therapeutic agent or therapeutic mechanism for a single pain condition or several pain conditions that may have the same underlying pathophysiology. A general therapeutic mechanism using a well-validated or approved pain medication as the biomarker probe with all samples from human sources. A novel therapeutic agent that has demonstrated robust efficacy using non-human samples for initial identification, followed by translation of the detection method in human samples. So that is important, that if you are doing non-human samples, then if it is in the UG3 phase, then the UH3 phase should have a translation that in the human samples. A non-pharmacological therapeutic regime that is generally acceptable or widely used in the treatment of pain. So devices or other non-pharmacological interventions such as acupuncture, etc., are also welcome, but those should be generally acceptable or widely used in the treatment of pain. Examples of responsive applications. So again, continuing to see this. So you can establish a single set of detection method components, including but not limited to from omics, imaging, or QST. Identification and optimization of the biomarker signature using a centralized resource, focus on clinical study coordination, detecting technology development, standardization of sample and data collection method, and statistical analysis and design methods, and optimization of the algorithm or signature to finalize the components and component for use in clinical trial design or treatment decision, and translation of biomarker signature developed in animals to detection methods in humans so that the biomarker signature is ready for clinical validation in a phase 2 clinical trials. So this is the translational if you have done in animals to translate into human. So example of non-responsive applications. So applications proposed to develop any biomarker signatures other than pharmacodynamic response monitoring or response prediction biomarkers. And natural history studies, they are not allowed. And the biomarker identification or validation proposing only animal studies without confirming use in the human, and non-standardized sample source selection methods, and having only large prospective design of clinical validation studies, therapeutic target validation and development, determination of efficacy and development of candidate therapeutics as the primary goal, and biomarker signature development for therapeutics that are not non-addictive therapeutics option for pain conditions. So opioids, if you are developing a biomarker for opioid therapeutics that are non-responsive here. Applications that do not include annual and UG3 to UH3 go, no-go milestone decision. And please note that non-responsive application will be administratively withdrawn. So you are encouraged to contact the program to know whether your application is responsive or not before you apply. So now we will move on to this scored review criteria. So specific to this RFA, so investigators, so knowledge and experience about the biological target and the disease biology is important. And the expertise in the target biology, pain condition, and the clinical phenotype, and whatever other modality you are using is important. Ability to manage the discovery process for the biomarker signature, and the you have to carefully define the roles of each collaborator in the team management plan, and description of the multiple investigator team if biomarker signature will be common across several pain conditions. And the review criteria approach for the specific RFA, a clear and realistic organization and execution plan for the identification and early validation of the biomarkers, strengthen the rigor in the data supporting rationale, including choice of therapeutic probe, and plans for the execution, plan for the execution and planning for the therapeutic intervention, including the choice of the therapeutic probe, and plans for ensuring standardization of data and sample collection, feasibility, and a carefully considered plan for the biomarker detection technology development, and the appropriateness and rigorous of the training and testing procedures of the machine learning if you are using that. And continuing with the approach, again, the bioinformatics and statistical analysis or deconvolution of the data clearly being stated how you will be doing, and plans to assemble a signature from several possibly multimodal components, and plans for initial proof of concept and validation and logical based on a clear understanding of the pain condition, plan for the development and validation of the detecting technology, and the innovation. Does the design or the research plan included is innovative elements is appropriate, which enhance its sensitivity, potential for information, or potential to advance scientific knowledge or clinical practice. So those are the review criteria specifically for this RFA. So as a general advice, your proposal should meet an unmet need. The proposal should be able to define what the current standards are and what the performance metrics, the candidate biomarker signature, and the methods of detection must meet in order to improvement over what is available as the standard practice right now. The study design should test the biomarker signature's utility for decision making within the context of use proposed. The statistical analysis plan should be clearly described and appropriate for the context of use. And the study design should consider and plan to identify and address source of variability. And the study section, so this will be reviewed by NINDS study section, and the reviewers come from a variety of expertise to evaluate different parts of the proposal. So don't assume that all the reviewers know everything. So you should be able to clearly articulate those sections in your application very well. So as a general advice, read the notice of funding opportunity carefully and schedule a call with the assigned program director prior to finalizing the submission, and provide a research plan that includes both the UG3 phase and the UH3 phase. Complete the milestones, timelines for each year of the grant. That includes both phases. Provide all information requested in the NOFO, including human subject protection, data sharing, intellectual property, team management. Provide a detailed budget and justification that is consistent with the research plan. And it is important to contact your program officer listed in the NOFO for any questions if you have in the above, or if you have any doubts, please contact us. And before going to Q&A, I just want to let you know that we have another approved concept, which is going to be very soon as a funding opportunity announcement. That is to developing remote or patient wearable device derived objective biosignatures or functional assessment to monitor pain. So this will be to address an urgent or unmet programmatic need, that is to develop and validate remote or patient wearable device derived objective biosignatures or functional assessment for use in the clinical trials and clinical practice as endpoints. So you can look into this into more details that's in the NINDS web page. And also that there's NINDS Biomarker Program. So these are the funding opportunities available in the NINDS Biomarker Program currently. So NINDS Biomarker Program supports development and validation of biomarkers and biomarker signatures for neurological disorders, including pain conditions that are within the mission of NINDS. So there are 3 funding opportunity available, and you can contact Dr. Taylor Birds. So Carol is the one leading the NINDS Biomarker Program. And you can contact, if you have any pain applications, or you can contact me or Carol, and then we are happy to talk to you about this. And if any questions, now probably we will be happy to answer. Thank you.
Okay, thanks, Ram. And again, anyone that joined in later, please submit your questions in the chat section below, and we're going to get to them now. So we have our first question, Ram, 1 second. Okay, our first question is, are biometric sensors and biosensor considered biomarkers when being used to measure inflammation from biopsychological skin measures?
Yeah, so those are biosensors and biosensor considered biomarkers when being used to measure inflammation. Yes, so they are biomarkers when you say that you measure the inflammation, but then what you are measuring, sensors are not biomarkers, and sensor development won't be, appropriate in this RFA. But then if you measure any biopsychological skin measures, that becomes the biomarker. And that is in the context of use. And remember that this is only for the prediction or the response biomarker for a therapeutics, not a diagnostic or prognostic biomarker.
Okay, next question. Is the HG3 similar to R61 R33 mechanism?
It's very similar. The only difference is UG3 UH3 is a cooperative agreement. I mean, like that is an NIH scientific officer will be involved in your study design if needed, and also mainly when you are defining your milestones. So R31, R33 is again the milestone driven program, but it is not a cooperative agreement with NIH.
Okay, next question. Can you apply for this RFA in the upcoming remote monitoring RFA?
Yeah, you could apply, but you could not submit the same application again. And also there should not be any overlap between those two if you submit for this RFA and the monitoring RFA.
Okay, have another one. It's quite long, okay. If the pain disease condition we want to study has no biomarker, can we propose a prospective cohort study to develop identification UG3 and validate the biomarker UH3 phase? And then it goes on to say, in your slide, it says large prospective design clinical validation studies is non-responsive proposal. Can you please kindly clarify?
Mm-hmm. Yeah, that's a good question. So large prospective design of clinical validation studies are not part of this RFA. And you could propose a clinical study to validate your biomarker, but not as doing as a prospective only for the purpose of doing a prospective design of clinical validation studies. Before you reach even to that stage that you are actually asking as a prospective cohort study to develop identification and validate the biomarker in the UH. That is allowed, but again, it depends on how you are designing your studies, and then we could be having a call to discuss that.
All right. In the context of response biomarkers, if an opioid treatment is a typical therapy used to treat a pain condition, should an opioid treated group be included in the study design?
If it is only the opioid treatment and your biomarker is the response biomarker for that particular opioid treatment, then that is not responsive and that is not the HEAL's mission. But if you have an arm of opioid therapy comparing to a non-opioid therapy and trying to develop a method or how to reduce the opioid treatment, then that is responsive.
Okay, that's it for now. We'll give it a couple more minutes. And again, the presentation and the slides will be shared early next week.
Yeah, for the last question that, so opioid sparing is welcome, but you should be carefully design the study when you have a opioid treatment is involved. So that should be evaluated as every proposal when it comes in individual basis. So I would encourage you to talk to us.
We can give it another minute, Ram.
Okay. Okay, thank you all. And if you have any more questions, please feel free to contact me. And then we are happy to help you in any way. Thank you all and have a great afternoon. Bye now. Is that okay, Rakanda? We will finish.
Yep, ending now.